Hello everyone, thank you for coming to this video. So the goal of this video today will be to put everything together and then explain basically the difference. You've seen the difference between phenotype and genotype in a previous video and presentation and all the material. Now what we're going to do, the goal of this video now is to explain the molecular mechanisms behind that, right? So we know that DNA is the instruction sets of a cell. Basically it's the blueprint for the life of that cell, right? So that cell is say a liver cell, and that liver cell does a certain amount of activities, things that the cell is specialized to do. When you think of a brain cell, for example, a neuron, well, this neuron has the exact same DNA as the liver cell if it's in the same individual. So it has the same set of instructions. However, the instructions are some instructions are set aside. They might be some actions might be done by the liver cells. The neuron has the same instructions but might not do any of that. Will not follow any of those instructions. It's going to remain silent. Other sets of instructions will be followed, and that makes a big difference between a liver cell and a brain cell. Both of them, when you think about it, have the exact same genotype. They have the same DNA. They're in the same person, but the phenotype of each cell is vastly different, very, very different. You think of liver cells, has nothing to do whatsoever with the neurons. They don't look alike, they don't live in the same environment, they don't have the same actions, they don't work in the same way, they don't consume the same nutrients. There's all sorts of differences between the two of them. However, the same genotype, but they don't have the same phenotype. So our goal today is to really look at what's happening when a cell has a certain genotype, but then what happens with the phenotype? How is the set of instructions? How does it translate into actions, right? And that's what we're going to look at. Now, when we look at the instructions of the instruction of a cell at a blueprint, then in this case, we're talking about DNA. And in our case here, being more specific, we're talking about genes, right? And now remember that genes are a piece of DNA. It's not the entirety, like it's not every single bit of DNA. A gene has a start and it starts somewhere and it ends at another place. It's a very specific piece of DNA, right? That's what a gene is. And of course, all along your 46 chromosomes, 23 pairs of chromosomes, there's lots of different genes there, and many genes are found on the same chromosome. It starts at here, it starts at a certain point, it ends at another point, right? So that's what you should know by now, that what genes are. Now, when we look at, so let me come back to this. So I mentioned something here about the blueprint. So blueprint basically is the plan, right? The plan. What is that cell going to do? What are the goals, right? So that plan is written in DNA language, right? So we have all the instructions are all stored in part of that cell, which is a nucleus. All the DNA is there. All the system instructions is there. Now that's the plan, but the plan does not lead to actions. We know that, right? So when you look at DNA and the blueprint and the plant, now we need at some point, will need to lead to some actions, and the cell will need to do something, whether it's digest a hamburger or send electrical signals so that you can lift something, right? So those are all the actions, and the actions of any cell is done via the proteins. So the proteins are actually the workers of the cells. This is where the action is done. It's done by all of those proteins, and one single gene, just one gene, will lead to many thousands and thousands and thousands of that same protein, right? So one gene, once activated, once the switch is turned on, this will lead to the production of many proteins. So this is a process that'll be going on and on and on and on until something switches the gene off again. So genes can be switched on and can be switched off. And there's very specific switches for that. But if I come back to this here, these are the workers or proteins, okay? Now, we have a problem here. And here's what the problem is. The problem is this. The problem is that DNA is written in a certain language. And that language, the letters or the vocabulary of these genes is written in A's, T's, G's, and C's, right? We know these are the nucleotide, and these nucleotides, there's 4 of them, and they're repeated over and over again in a sequence that forms DNA. So basically, that is how DNA is written. Now, when you look at proteins, well, proteins are not made of nucleic acids or nucleotides. Proteins are made of amino acids. Amino acids. And we know that there are 20 different types of amino acids. And what is the relationship between these guys and these guys here, nucleotides and amino acids? The relationship between them is 0. There's no relationship between the two of them. We need something in between that is able to translate this into this. We need a piece of equipment right in the middle here. And that piece of equipment here needs to be able to crunch those nucleotides and then make sure that the cell has the workers, the proteins, to do the action. And this is what we're going to look at. Okay? So we're going to look at this whole process here, which is the start line right here, and all the process down to here, which is the finish line, which is obviously producing our proteins. Okay? So that is that's an important part, and this is what we will be looking at. Okay? So let me look at this again. Okay? We have our DNA, and then at the end we have proteins. All talk, no action. Action. Okay? So in the middle, we have this missing piece here. And that missing piece is that part that can relate to both DNA and the protein at the same time. And this is called the mRNA. mRNA for messenger RNA. And RNA is another type of nucleic acid that is related to DNA. But one is ribonucleic acid, and DNA is deoxyribonucleic acid. So basically, it's just a little bit of a transformation on each of the nucleotides. They look very much the same. One has, if you look at the subunits of DNA and RNA, you'll see that RNA has a sugar called ribose. And then if you look at DNA, you'll see it has deoxyribose. So it's just a change in the sugar structure. And I really invite you to look at the monomers and understand what the difference is. It's very, very slight. It's a slight little hydroxyl group that makes all of that difference. Now, when we're looking at DNA, and here, of course, I'm talking about the genes, right? I'm not talking about the DNA that is not part of does not contribute to the phenotype. So of course here we have a part, and that part is, we talked about that before, genotype, right, it's your genes and everything, and then here, because this is where the action is, here we have the phenotype. Okay, genotype and phenotype. So proteins are responsible for the phenotype, genotype is DNA, and most importantly the genes, right? So we have this thing here in the middle part, which is the mRNA, and this mRNA is going to be responsible for translating this genotype into a phenotype. So basically it's telling the cell, this is what you're doing now. It might even tell that cell, you are a liver cell, you are a brain cell, you are a heart cell, you're a muscle cell. Okay, all of that based on all of this here, okay? So this is what the important part. Now, this here has a name, and it's called the central dogma. Central dogma biology, in biology. And this dogma has been transformed a little bit over the years, but basically it states this. It states that DNA is transformed into RNA and then is eventually transformed into proteins, okay? So that's the important part here. When we look at a cell, we have the structure. So here, of course, I'm talking about a eukaryotic cell, a cell that has a nucleus. Remember that prokaryotic cells do not have a nucleus. We're not going to talk about that. We're going to concentrate on the eukaryotic cell right now, okay? So we have a nucleus inside a eukaryotic cell, and inside that nucleus we have our DNA, right? We know that DNA is sequestered inside the nucleus, and it never really leaves, okay? So this DNA will be transformed by enzymes. It will be transformed inside the nucleus, that's important, inside the nucleus, into an mRNA, messenger mRNA. Now this mRNA is going to be slightly modified inside the nucleus. It's going to go through a maturation stage, and during that stage, eventually at the end of that stage, our mRNA is going to be exported out of that nucleus, okay, out of that nucleus. Inside the cytoplasm, once the mRNA has moved from the nucleus to the cytoplasm, outside of the nucleus, the mRNA is going to be taken in charge by ribosomes. And the ribosomes are going to interact with this mRNA and are going to transform this mRNA into proteins. Okay? So, there are two processes here. This process here is different than this process here. So basically the DNA to RNA has a name, it is a process, and then the other one here is another process. Okay? So this process here is called transcription. Transcription. So basically, it's as if somebody's reading a book in one language. They're transcribing these letters into another kind of alphabet. Okay? This is not a translation, it's just being transcribed. Okay? It's just changing the letters, but we don't know what they mean, those letters. That's the other job after, which is the job of this process here, which is translation. Okay? So, the transformation of an mRNA into protein is called translation. The modification, or I shouldn't say it like that, the reading of DNA and synthesis of mRNA is called transcription. All right? So, what is this about? Well, DNA has those letters. So we have A, T, C, and G. And we know that this is the normal pairing rules for DNA. For RNA, because RNA is also nucleic acid, rules are very, very similar. However, there is a main difference. So, A will be paired with a U, which is a molecule that's very, very similar to thymine. It's called uracil. It's very similar to thymine or the T. The base pairing remains exactly the same. A and U, 2 hydrogen bonds between the two of them. U, A, C and G still remain, and then G's and C's, right? So that's our RNA language, the letters. Not the language, but the alphabet. And don't forget that RNA, most of the time we talk about RNA as a single strand, DNA double-stranded. RNA can also be double-stranded, so therefore that's why I explained this part here, okay? The other thing that's important is that RNA also has a 3' to 5' end, okay? Just the same way our DNA has 3', 5'. Well, RNA also has that. That's an important part. Now, when we're looking at proteins, we have amino acids, as I mentioned earlier. And of course, there are 20 different amino acids. And the combination of all those 20 different amino acids will lead to the production of different proteins, almost an infinite amount of protein. Not really infinite because our cells are limited, but the combinations could be almost infinite. There's a huge amount of combinations. So, that's what we need to understand now. Now, what we're going to look at for the next segment of this video is this process here. So basically what we will focus on is the transformation of our DNA into mRNA. How is this done? Okay? And then, after that, we're going to look at this transformation right there. So this is what we'll be focusing on for the next segment of this video. We are going to look at this mechanism here as I said earlier. So what we're going to look at right now is the transcription. So transcription process is made possible by an enzyme, a very important enzyme, and that enzyme is called RNA polymerase. So remember that when we replicated DNA, we used an enzyme called DNA polymerase 3. We also had DNA polymerase 1. If you look at the mechanism, it's used later in the process. But DNA polymerase 3 is an enzyme that copies DNA. Well, RNA polymerase 1 makes a copy of one strand of DNA. One strand of DNA. So DNA has 2 strands, anti-parallel strands. One of those strands will be used, and then the RNA polymerase is going to read that strand and transform this strand into RNA. That's why it's called RNA polymerase. It's going to polymerize an RNA molecule. Polymerize because it's a polymer of RNA ribonucleic acid or ribonucleotides monomer for RNA. Well, this is what we're going to look at. Now, when we're looking at DNA, remember that DNA has an orientation, anti-parallel orientation, 3' to 5', 3', 5', 5' to 3' on the other side. Remember that they are always anti-parallel. Now, mRNA, I said earlier that RNA can be double-stranded sometimes. Don't worry about that. This is not what we're talking about now. The mRNA is single-stranded. It has only one strand, but of RNA. It's not double-stranded, but it does have an orientation too, 3', 5', right? The molecules are, the monomers are very similar to DNA, and therefore the end, when you look at this RNA here, if you look at that end, it's very different from what you see at that end here, okay? It's still RNA, but depending on where you look at it from, this side or this side, then it has a different look, and enzymes of course recognize a piece of it and they don't recognize the whole molecules as a whole, but they recognize small pieces of it, and in this case, the ribonucleotides. So what we're going to look at right now is the process of transcription. So now one thing that I need to tell you is this, is that on DNA, in order to identify where the genes are, remember the genes are part of a huge DNA molecule, and there are smaller pieces of it. They're interspersed all over the place, right? And these genes, in order to be able to recognize exactly where are they, the enzymes such as RNA polymerase has a way to detect those genes, where they are. And of course, if we wanted to, we can go into the molecular biology of this and explain all the small details. It's quite a complex process. This is not where I want to go right now. I want to explain the basics of transcription, and that's what we're going to do. Okay, so you can read and look at all the small details, and of course in the videos I posted on D2L, you will see that some of those processes are vastly complicated. Don't get lost into those complications. This is not that important. What is important, though, is to understand that the genes need to be recognized by the RNA polymerase, right? So this portion here is some kind of like a marker that says, here's a gene right there, and this is where you should be starting. So this part here is called a promoter. The promoter is a part of a gene that is usually situated upstream of that gene. So what it means is that past this way here, this is upstream here, downstream where the enzyme is going to read, upstream from there, from here, we have the promoter. Okay? So the promoter will allow the interaction of the RNA polymerase with DNA. Okay? So, for the next part of my video here, I want to draw what the process of transcription is. Okay? So what we have here, say that we start with a DNA molecule from 5' to 3' to 5', and then on the other side here, of course, we start 5', and then we have our promoter right here. Okay, so when the promoter attracts the RNA polymerase, the RNA polymerase will interact with this promoter right here, and will basically sit on that spot right. So 5' here, this molecule is going to sit there, right? And on the promoter, that's right underneath, right there. And then what it's going to do is that it's going to melt this DNA. So basically open it up, okay? So again, 5' right here. And then what we're going to have is we're going to have the enzyme RNA polymerase is now going to interact with that piece of DNA right here. Now, the direction of synthesis is this way, right? So basically what it's going to do is that it's going to create a molecule here that starts at 5', and then this molecule, this RNA polymerase, is going to read every single nucleotide here, right? And out of that RNA polymerase, we're going to have our mRNA that's going to come out of that. Basically what it does is that it reads one nucleotide and it adds the nucleotide that is corresponding to that nucleotide, right? So for example, if it reads a T, it's going to add a U, because remember that's in DNA. In RNA we'll have a uracil, right? So if it reads a T on DNA, it's going to add a U. If it reads a G, it's going to add a C, and so on and so forth. So of course, as the RNA polymerase moves onto that piece of DNA here, and that piece of DNA by the way, it's called the template strand. Template strand. And that template strand is the one that is being used and read by the RNA polymerase. So the RNA polymerase is going to move on to that on that DNA strand right here, right? And as it moves on, it produces this mRNA that we see right here, mRNA. And you see here, it creates this this bubble. Remember? Okay, so remember the replication bubble for DNA? Well, this one is called a transcription bubble because the DNA is temporarily disconnected from the other strand. So what the RNA polymerase is, RNA pol, so let's say R-pol, and R-pol is moving in this direction right here, and it's creating this molecule here that now has so this is our 3' here. So basically what it does is that it creates a 5' molecule here and a 3' molecule here. And that will lead us to the formation of this mRNA here. Now, one thing that you need to understand is this. When we look at those strands, so let's say that I have my template strand, right? So template, and let's say that my sequence for my template strand is this. Let's say that my template strand has this sequence here. Okay, so let's say G, C, A, T, G, G, C, T, A. Okay, that's my template strand. My template strand is the 5' to 3'. Okay, my RNA is going to have this sequence. Is going to have the opposite sequence of this. So it's going to have C, G, U, remember, right? Yeah, and then we're going to have A, we're going to have C, C, G, A, and then U right here. So you see this is complementary, right? So the template strand and the mRNA have a complementary structure. But if you look at the other strand, that's on top of that DNA, the 3', 5' here, that is the non-template strand. So this was the template strand, this one is the non-template strand. The non-template strand, its sequence is C, G, T, A, C, C, G, A, T. So now besides the U's, the whole sequence is exactly the same, right? So what is important here is that, you know, the mRNA has a sequence that is complementary to the template because that's the RNA polymerase, that's what it looks at. It looks at the template, and the only way it has to create an mRNA is to look at the bases of the DNA and then put the bases are complementary to that. But now when you look at the mRNA, you can compare that with the non-template strand and you know, well, hey, it has exactly the same sequence except in RNA language, okay? So that is the part of the transcription, okay? So of course, like I said earlier, this process can be highly complicated. We can bring all sorts of other proteins that are interacting here, and there are many of them, but we don't need to go there, okay? At least not for this part of that course. So, but this is important, okay? So I wanted you to really pay attention to this, and then we're going to move on to protein synthesis. So now our mRNA is now made, is synthesized. Now what we can do is move on to translation part, right? So we were successful in transforming DNA into RNA or mRNA. We know how that works. We know that it reads at one of those two strands. That's called a template strand. We know that it creates an mRNA that is the opposite image, basically the complementary image of that DNA, the template strand. We know that it proceeds in a certain direction, right? So it reads the template strand in a certain direction. Of course, it needs to be oriented properly. And then we have now created this mRNA. And don't forget that this mRNA does have an orientation. Now, this mRNA is special because it is coded in a certain way. And every 3 nucleotides on the mRNA, so of course you can imagine it'll be very long stretch of nucleotides, but every single one of those nucleotides is actually paired up with 2 other nucleotides. So it creates this trio of nucleotides, and that trio is called a codon. So we have a codon right here, and that codon has a certain base organization, right? So let's just make one up. So let's say it has GC, right? Okay, so that's just a sequence. And that's codon number 1. And then next to it, there's codon number 2, there's codon number 3, 4, etc., etc. And every single one of those codons might be exactly the same combination here, or we can have a different combination, right? All the other nucleotides, it can be a lot, we can have up to 64 different combinations for these codons, okay, up to 64 of those combinations. And all of those codons here will now be recognized by a process that will, by a machine, really, it is really a biological machine, that is going to read this information here in the mRNA and transform that into protein language or amino acids, okay? So that will be the process of translation. The process of translation is a complicated one. And like I mentioned, what we need in order to be able to be successful in this process is a biological machine that is called a ribosome. Now, one thing that I need to mention here is that in this video lecture, I am not going to spend some time explaining how the ribosome do all the reading on the mRNA. And the reason being is that there are very good videos that I posted on D2L, very good videos depicting this. They explain in very good details and visually appealing videos that will explain much better. There's animation that do a much better job than what I can do on a board like this, right? So this is why I decided not to explain everything that happens when a video can do so much more than what I could do here. But the thing that I need to tell you is this, is that a ribosome is actually made of 2 subunits, and they basically would look like something like this, and that would be the big subunit right here. We have 2 subunits, 1 subunit here, and a smaller subunit. So a large subunit and a smaller subunit here. And what the ribosome does is that it will read, so if I have all my codons are organized here, right, all my nucleotides here, and every single one of those nucleotides are organized into codons, trios of nucleic acids, right? So we have 1, 2, 3, 4, 5, 6, and 7, etc. The ribosome is going to read and stop every time at every single one of those codons, and will decide this codon here is codes for amino acid number 1, and then amino acid number 2, and amino acid number 3, etc., etc. In order to know what amino acid will be added, then we needed a genetic code for that. And in the book, in the chapter, there is an illustration, a table that tells you exactly what the genetic code is. And of course, in the videos, that this will be explained as well, the videos that I will be posted on D2L. But what you need to know is that the ribosome moves on at warp speed very, very quickly on this mRNA. And then there is a pocket somewhere where the protein with all the amino acids are going to be added one after the other, and the protein is going to come out of that ribosome. And of course, again, this is very well illustrated in the videos that I will post on D2L, okay? So I don't want to go too much into the details of this. I just wanted to give a very broad, big picture of what's happening here. This you will see, and but it's important to understand how all of that works, okay? So thank you.